Okay, that looks good. Welcome, folks. This morning's evolution will be Regina Fairbanks from here at Davis talking about tga1. And Regina, if you're ready, take it away.
Great. Thanks everyone for coming to hear about my work so far on tga1 evolution, which has been a kind of confusing but interesting project to work on. So many of us probably already appreciate that domestication is one of the most important and interesting developments of the last 10,000 years. The domestication of hundreds of plant species worldwide catalyzed the independent origins of agriculture around the world, which radically altered the shape of human history and the entire planet. And from an evolutionary perspective, domestication involved the adaptation of wild plant species to cultivated environments and human-mediated reproduction. And as we especially appreciate, domestication sometimes resulted in crops that looked so distinct from their wild relatives that sometimes botanists struggled to figure out who their wild relative even was. So today, archaeologists and evolutionary biologists have both made a lot of progress in understanding domestication, especially where, when, and kind of how it happens. And I'm thinking a lot about this major topic in domestication, where we're really interested in understanding what the nature of selection was like during domestication. So people have been interested in things like how strong selection was, especially how it compares to selection in natural populations, and whether selection occurred primarily on new variation that was popping up in cultivated populations, or whether domestication was primarily pulling from standing variation that exists, segregating in wild populations pre-domestication. And similarly to that, people are comparing these patterns between different crops. So in maize, some early researchers thought that only selection, mostly on like new novel variation, could explain the dramatic differences in morphology between teosinte and maize. But over the last few decades, evidence is kind of increasingly showing that it's the opposite pattern that's true, where maize domestication drew mostly from standing variation, so variation that's segregating in wild teosinte populations, rather than de novo mutations. So I have some examples here of important genes underlying domestication traits, many of which have also shown signals of selection. And these are ones where the domesticated allele, the allele that's in maize, has also been found in teosinte populations. And these are for some really important traits like plant branching and how many ears maize has. So these few genes suggest that selection for these traits during domestication drew mostly from pre-existing variation in wild teosinte populations rather than new variation that arose in cultivated populations. And as we've learned more about teosinte, this makes sense because we know that teosinte has a lot of genetic variation, and field surveys can often find maize-like phenotypes in wild teosinte. So it's increasingly seeming like maize domestication pulled a lot from standing variation. There is thought to be one exception, and that's what I'm going to be talking about today. And this is tga1 and its associated domestication trait, exposed kernels in maize. So as many of us know, one of the most notable changes during maize domestication has been the loss of the protective fruit case. In teosinte, the kernels are all covered by this tough inedible fruit case, which are thought to protect the seeds from seed predators. And during domestication, maize lost the fruit case, which resulted in these fully exposed kernels. And these exposed kernels are thought to be really deleterious in the wild, but they would be advantageous in cultivated populations where human farmers would have protected the plants from seed predators, and also presumably really prefer the exposed kernels because they're a lot easier to process than their fruit case and enveloped relatives. So tga1, and actually more specifically one single mutation in the gene, seems to control a lot of this phenotypic difference in fruit case exposure between teosinte and maize. So work around 10 and 20-ish years ago now found that when the maize allele of tga1 is introduced into a teosinte background, you lose the really tough inedible fruit case, and now you have these papery coverings that don't fully enclose the kernel. And on the other side, though the effect is less dramatic, you still get, when the teosinte allele is in a maize background, you start to get harder tissues that start to cover more of the kernels. So this seems to be the major gene that controls the exposed kernel phenotype. So the current understanding of the origin of exposed kernels and similarly the evolution of this domestication gene tga1 is something like this, where exposed kernels arose very shortly after people started cultivating maize, or what would have been teosinte at the time. And then people, as soon as exposed kernels appeared, exerted strong selection on that trait. And then because of that, exposed kernels really rapidly fixed in these early domesticated populations. So far, though, the evidence supporting this, what we're going to call a de novo origin model, followed by strong selection, have been somewhat conflicted. And one major reason is that that people are pointing towards this de novo model is that, unlike for the other canonical domestication traits, field surveys haven't really reported exposed kernels in wild teosinte populations. And this is thought to be consistent with assumptions that exposed kernels would be really highly deleterious in wild populations, since those plants would be really susceptible to seed predation, and that trait would be quickly removed from the population. But if exposed kernels did experience really strong selection right after appearing in cultivated populations, we would expect to see signatures of selection on the genes underlying this trait, and most importantly on tga1. But tga1 often fails to appear in scans for signatures of selection in maize genomes, which is part of why we started to think that maybe this model of a de novo origin followed by really strong selection was not quite accurate. So the question that we've been asking is tga1 really this exception to what we're starting to think is a general pattern in maize, where the domestication traits and the alleles underlying them are being pulled from standing variation in teosinte? And I have been trying to answer this question using mostly population genomics approaches with the ZMAP dataset of modern maize and teosinte genomes. So this has about 500 inbred maize genomes and about 150 wild Parviglumis and Mexicana genomes to see whether I can better understand the origins and evolution of tga1. So before jumping into what I actually did, I'm just going to give some important PopGen background about selective sweeps so that we're all on the same page about how these methods are working. So selection does leave behind signatures in the genome as a result, mostly of this process referred to as genetic hitchhiking. And I'm going to illustrate this process with this little schematic here, where we're starting out with this population that has 6 different genetic backgrounds, and each has some neutral variation, which I have represented as these white circles. And here a beneficial mutation arises, which I have as the new teal circle, and as soon as it arises, it's this de novo mutation, it's immediately selected for. And the selection causes it to rise in frequency in the population, but you'll notice that not only has that beneficial mutation arise in frequency, but so has the neutral genetic variation that was nearby that beneficial mutation. And since there's strong selection, the mutation is rapidly rising to fixation, and there's not a lot of time for recombination to occur. Maybe some small amount does, but in general, that whole genetic background gets swept up in frequency alongside the beneficial selected mutation. So now, once the mutation has fixed in the population, we see that the genetic diversity near the beneficial mutation has decreased, and that pattern of reduced variation near a beneficial mutation is what we're detecting when we look for signatures of selection in the genome. And this scenario that I've described is referred to as a hard selective sweep, and is what we would expect if tga1 arose as a de novo mutation and was immediately strongly selected for. So to understand what this haplotype diversity patterns would look like from a hard sweep followed by from a hard sweep from de novo mutation, I simulated maize domestication in SLIM, and I'm going to describe that in more details later. But for now, I'm just going to show some simulated haplotype diversity visualizations to get an idea of what we might expect in our real maize data. So I'm going to show the plot in the next slide, but just to orient everyone, each row is going to be an individual haploid genome, and each column is a different SNP within a 10 kilobase window surrounding the mutation that was selected for. And then in the plot, each black tick is going to represent a derived allele at that SNP. And for the simulated plots, an individual haplotype is just randomly selected, is at the top of the plot. And then as you go down, the haplotypes are more different from that top haplotype. So what we're seeing here from the hard sweep is that there's basically kind of a shared giant haplotype for this entire 10 kilobase block surrounding the mutation. And since some time has passed since the mutation fix, there's some like lower frequency variation that has appeared and kind of drifted around in frequency, which is causing this little scattered pattern that we're seeing. And before showing the real maize data, I also want to consider our alternate hypothesis that the maize allele of tga1 existed as standing variation in the ancestral wild teosinte populations. So selection from standing variation will also leave behind genetic signatures, but these will be distinct from those left by those from hard sweeps or de novo mutations. And using a similar schematic as before, here the mutation has actually existed some time in the population, and it's kind of floated around as neutral variation, and it's had time to recombine onto different genetic backgrounds than the one it rose onto. And by the time selection starts in our population here of our 6 like little fake haplotypes, the mutation now exists on several genetic backgrounds. And when the environment changes and the mutation is now beneficial and selected for, each of these haplotypes will increase in frequency along with the mutation. So what we end up with is a final population that has reduced variation around the mutation, but it's not as dramatic of a reduction compared to the hard sweep model. So I also simulated this scenario where a mutation starts in our population at 1% frequency, and we see a really different pattern. So instead of almost all of the individuals sharing basically the same haplotype, except for some scattered variation that appeared since the mutation fixed, we now have several distinct haplotypes that are kind of these chunky barcode blocks. And in our data from modern maize, we see a pattern that's kind of between the two, and this will kind of be a theme throughout what I found, where it's kind of confusing what kind of selection was happening and how the mutation arose. But here the haplotype that's at the top row is B73, and each haplotype plotted below is increasingly different from B73, with the bottommost haplotype being the most different. So the first thing that's obvious is that we don't see a lot of variation at all compared to our simulated data, and this might just be how I was simulating things. So that's why you're seeing fewer SNPs plotted for the same kilobase window, 10 kilobase window around a beneficial mutation. But something I want to point out, because these are not super intuitive to look at, is that we're noticing kind of 2 different haplotypes, at least at moderate frequency, maybe more than 2. And this at least is inconsistent with the hard sweep model, where we would expect really just one dominant haplotype with maybe some scattered variation that appeared since the mutation fixed. So our takeaway from visualizing the modern maize haplotype diversity is that it doesn't seem to clearly support the prevailing model, which was a de novo origin followed by a really strong selection for the causative tga1 allele that makes the exposed kernels in maize. But it also doesn't obviously resemble the diversity patterns I was getting from simulating a sweep from standing variation. So with my SLIM simulations, I wanted to explore not just the de novo versus standing variation model, but a couple other scenarios that could produce greater levels of haplotype diversity in modern maize. So previously, it has been hypothesized with some support from early modeling work in that 2005 paper that selection on tga1 was really strong. And this makes sense because we think of maize as a grain crop, and exposed kernels really allow maize to be used as a grain crop. But if we imagine a scenario where selection is weaker, the mutation's frequency will increase more slowly. It'll rise to fixation more slowly, which leaves more opportunity for recombination to shuffle that beneficial mutation onto other genetic backgrounds. So what we end up with as a result is a bit more genetic diversity around the beneficial mutation than we would expect from a hard sweep that involves really strong selection. Another scenario that could produce greater than expected haplotype diversity is if recombination is a bit elevated at tga1. And if this is the case, there could be more opportunities for even a de novo mutation experiencing strong selection to be shuffled on to other genetic backgrounds early on during the selective sweep. So that would also cause multiple haplotypes to rise to like moderate frequencies in the population. And we do have some reason to think that recombination might be a bit higher at tga1. I did an analysis with LD helmet, which found that the region right around the mutation in tga1 has a recombination rate that's about 10-fold higher than the chromosome-wide average, but we weren't quite sure whether that alone could account for the haplotype diversity patterns that we observed in our actual data. So to evaluate these possibilities, I used SLIM simulations to model the evolution of tga1 during maize domestication. And I first started with coalescent simulations of a kind of ancestral teosinte population to produce some standing variation that I could feed into the SLIM simulations. In SLIM, I then modeled the onset of domestication, which here I included a population bottleneck with a dramatic reduction in population size. And at this exact generation is when we start selecting for the mutation in tga1 that's beneficial. For the de novo origin simulations, I introduced that mutation right at this generation. And for the standing variation models, I randomly selected an existing variant and just changed its selection coefficient to change it from neutral to beneficial at this generation. And then the remainder of this simulation was basically exponential population growth of the maize population until present day. Here, the demographic parameters I use are just adapted from those specified by Basinger et al. In their 2016 paper. But because SLIM can't handle really large population sizes, I had to downscale them all 10-fold and accordingly rescale the generations, selection coefficients, recombination rates, and mutation rate. And it's those rescaled values that I'm going to be showing on later slides. And so once I had simulated all of this genetic variation, I then calculated some statistics that are used to understand haplotype diversity, and these are H12 and H2H1 statistics. And these were developed to detect selective sweeps, but also to differentiate between hard and soft sweeps. And I use those to kind of assess how similar the simulated results are to the actual data. And the last thing I want to note is that these are kind of really simple simulations. I'm just accounting for the bare minimum that we think is important, which is basically a population bottleneck and then exponential growth of maize. So these are more coarse estimates of what parameter sets kind of make sense to explain modern haplotype diversity patterns. So I simulated 3 different mutation starting frequencies. So this is a de novo origin where it appears in just one individual. I have the mutation at 1% frequency and also at 5% starting frequency. And then I used 2 different recombination rates. And then I actually ran 5 different selection coefficients, but only 3 are shown here. And this is because we know that tga1 is fixed in modern maize. So I've only plotted those parameter sets for the mutation fixed by what was modern day in the simulation. And I found that only strong selection coefficients resulted in a mutation that fixed by present day in the simulations. And for each parameter set, I ran 1,000 replicate simulations and then calculated those H2H1 statistics on the windows that contained the selected mutation. And I'm going to be showing those plots in the following slides. And I'm also going to point out that to compare those simulated statistics to those from real modern maize, I have those real values from modern maize as dotted lines. So the first thing I'm showing is the results from my de novo simulations, where I found that only those simulations with the absolute strongest selection fixed even more than 10% of the time, and even these fixed only about 40% of the time. So this is the strongest selection coefficient that I ran. And you'll notice that of those, most of them don't produce H2H1 values that are near those we calculate for modern maize at the beneficial mutation. And this is pretty consistent with what we were observing from the haplotype diversity visualizations, where a de novo model doesn't quite seem to explain the elevated haplotype diversity that we see in modern maize near tga1. Like the de novo simulations, the mutation didn't fix with weak selection, even in these standing variation simulations. But the mutation did fix with slightly less strong selection, in contrast to the de novo simulations, where only the absolute strongest selection coefficient allowed the mutation to fix. And even more interesting is that unlike the de novo simulations, where it fixed like not even half the time, here the mutation fixes almost all of the time with the strongest selection coefficient and more than half the time for the slightly weaker selection coefficients. And this makes sense because if you're starting from a higher frequency, you should be able to fix more often than a de novo mutation, which could get lost to drift early on. And what's also notable is that unlike the de novo simulations, where they basically never produce H2H1 values that are similar to those we obtained from modern maize near the beneficial mutation, we do see that more of these runs are able to produce H2H1 statistics that are similar to those we get in modern maize. And this is true for either the 1% or 5% standing variation runs. And in general, across all of these runs, it doesn't seem that recombination rate can explain the haplotype diversity patterns we see in modern maize. So it looks like recombination rate doesn't matter. Selection coefficient does matter. It should be pretty strong. And the standing variation simulations seem to produce H2H1 haplotype diversity statistics that are more similar to what we're seeing in modern maize. So if we're thinking now that maybe it's a standing variation model, we would expect that the mutation might persist in modern wild teosinte populations. And because the allele is co-dominant with the ancestral teosinte allele, we would expect to see at least partially exposed kernels to be visible to collectors doing field surveys. But as I mentioned previously, field surveys have recorded an absence of exposed kernels as standing variation in wild teosinte populations. And what this means is that people have seen exposed kernels in wild teosinte, but they attributed them to recent gene flow from maize rather than using that as evidence for standing variation. And just as a reminder, it's just widely assumed that exposed kernels are just too deleterious to persist in teosinte for many generations. So we just wanted to see, like, are there any individuals that in our large sample of 150 teosinte individuals from the ZMAP dataset, are there any that have maybe the maize allele? And this is just showing that it's just one base pair difference where teosinte has the G and then maize has a C at this same spot in tga1. And what we find where I have this kind of haplotype plot where the maize is the teal again and the teosinte is the orange. And this star right here is that mutation spot in tgi1 where all of the maize is fixed for the derived allele, and then you see a couple teosinte alleles that also, teosinte individuals that also have the maize allele. So this seemed like this is evidence for the first time that there is maize tga1 alleles found in modern teosinte, but this isn't necessarily evidence for the standing variation model, because just because I identified the maize allele in modern teosinte doesn't mean that the allele originated in teosinte and would have been present as standing variation when domestication started. Because like I said, ongoing gene flow between teosinte and maize could have resulted in an introduction of the allele into teosinte from maize. So to test this, we decided to look for evidence of admixture across chromosome 4 in these heterozygous teosinte individuals using this phylogenetic weighting approach. And this approach basically just makes phylogenetic trees for all of the samples of interest, which are some maize, Parviglumis mexicana, and the heterozygous teosinte individuals. And it makes these trees in sliding windows across chromosome 4. And then I computed these topology weighting values, which basically represent how often our sample of interest clusters with maize individuals versus how often it clusters with teosinte individuals. And if it clusters with the maize individuals, we would interpret that as evidence for maize ancestry at that window. And for comparison, I've plotted what that looks like for some pure maize individuals versus for some pure teosinte individuals. And you'll see that on the x-axis, we have position. This is just a window around tga1, where the dotted line is the causative mutation that leads to exposed kernels, just to highlight where that position is. And on the y-axis is the topology weighting values, which we're just kind of using as a proxy for ancestry proportion. And for these pure maize and teosinte individuals as examples, you'll see that they look pretty different just upstream of the causative mutation. And for the heterozygous teosinte individual, they do more closely resemble the pure maize individuals right around tga1 compared to the pure teosinte individuals. So we took this as evidence that these heterozygous teosintes got the maize allele from some earlier gene flow with maize. And we did some kind of back of the envelope calculations based on the size of the block that seems to be of maize origin. And we estimated that gene flow from maize probably occurred hundreds to thousands of generations ago. So while this finding doesn't support our standing variation model because it suggests that the maize allele hasn't been in teosinte populations since it first appeared, but instead has been more recently introduced from maize, it does suggest that the maize allele can persist in wild teosinte populations for hundreds or thousands of generations, which counters previous assumptions that the allele was just too deleterious in the wild to hang around in wild teosinte populations for any substantial amount of time. And the last thing I want to note is that we do have like a relatively large sample size of teosinte genomes compared to some previous studies, but it's still a small enough sample that there's a reasonable chance we could miss a really low frequency allele. So we have 70 parviglumis genomes, which is the subspecies that's more closely related to maize. And so that's 140 haplotypes. And we would expect to miss an allele at 1% frequency at a rate of about 25%, which is kind of high enough that we might not want to take the lack of evidence for maize tga1 and teosinte as really strong proof that it doesn't exist as segregating variation. The other thing that we were thinking about is if this allele was standing variation, the mutation that causes exposed kernels should predate the onset of cultivation if it was segregating variation in the ancestral population. But what we find is that we actually get similarly contradictory results from this mutation age analysis. We used Relate, which is a software that estimates genome-wide genealogies to estimate the age of the causative mutation in tga1. And we get an age that's basically around the time that cultivation probably started, which is actually what we would expect from the de novo origin model. So in light of the haplotype diversity statistics in modern maize, this is kind of confusing because we were starting to think that a standing variation model explained things a little bit better, but now we're getting this age estimate that actually supports the original de novo origin model. So I'm just going to wrap up a bit, and our conclusion for now is still kind of tentative and confusing because our results provide evidence for both of these conflicting models for the origin of exposed kernels via this mutation in tga1. So on the one hand, my SLIM simulation suggests that a de novo origin of tga1 couldn't really produce haplotype diversity statistics similar to those we observe in actual modern maize data, and instead a standing variation origin with relatively strong selection seems to produce similar haplotype diversity statistics to modern maize. And we also found some evidence that maize tga1 can persist in teosinte populations for some hundreds to thousands of generations, but these modern teosinte populations only acquire that allele through gene flow from maize, not that the allele seems to have originated in the ancestral teosinte population and hung around since then. But with that said, maybe some more extensive and complete sampling of populations near the domestication center could potentially find the maize allele in the future. And then we also don't find evidence that the mutation is older than cultivation. Instead, we find that it seems to date to just around when cultivation started, which supports a de novo rather than standing variation model. So kind of confusing results. And looking forward, I want to think a bit more about the general patterns of how important standing variation was in maize domestication. So I'm going to be calculating some selection statistics across the maize and parviglumis genomes, which will allow me to detect some signatures of selection for completed or nearly completed sweeps. And then after I have those sites that are under selection, I'm going to be looking at how often those selected alleles exist in our modern maize data to have a more broad understanding of the relative importance of de novo versus standing variation during maize domestication. So thank you all for tuning in, and I want to thank the lab and some funding sources, and also the 10,000 years of farmers who have cared for maize and cultivated it for many years. All right, thank you.
Thanks, Regina. We have plenty of time for questions. One already from Marcus in the chat. Have you considered multiple functional mutations or the same mutation on multiple backgrounds?
In general, we have thought it's unlikely that the same base pair is mutated multiple times to the same from G to C multiple times, but I'm not sure how likely it is to be multiple origins.
Marcus, were you thinking multiple distinct mutations rather than multiple mutations at the same site?
Well, I guess you assume multiple distinct mutations. Some molecular biologists would have probably figured it out by now, so maybe not that one, but potentially that one. But when looking at the haplotypes, you showed it yourself, there's like those two groups. And those two groups, I mean, could be just twice the same mutation.
So you're thinking since domestication, the mutation has occurred twice on two different backgrounds? Or
yeah,
in
maybe. I mean, it does seem, given mutation rates and stuff, it does seem less likely, but I mean, by chance could happen, maybe.
Regina, have you thought some about why? I mean, your data suggests that really strong selection seems implausible in terms of we're unlikely to see the diversity statistics that we see in tga1 and modern maize. What I'm struggling a little bit with what are the implications for domestication if we think tga1 wasn't under crazy strong selection?
Yeah, I think a lot of the assumptions that tga1 would have been strongly selected for, and like exposed kernels would have been strongly selected for, is if people early on were using maize as for the kernel as a grain crop. But people have also proposed that early uses of wild teosinte and cultivated populations would have been for the sugary stalk. If they were mostly using it for the sugary stalk or for other purposes than the grain, then selection wouldn't have been super strong on exposed kernels early on, and it might have actually been like deleterious, because some people have speculated that exposed kernels are more susceptible to fungus and other things. So that's one implication if selection wasn't super strong.
And why do you think there might not be strong selection in teosinte?
It might just be not as deleterious as people have thought it was. I'm not sure if we know a lot about the actual ecology of how often teosinte is, like how often seed predators affect teosinte and how strong of a pressure that might actually be.
Is it possible that there's strong selection on some populations of teosinte and not others, like if seed predators were an issue, or even in cultivated maize, like strong selection on some, I don't know what to call that, cultivated teosinte maize things? like maybe some areas there would be incentive for that to be like a more complete casing around the seed.
Could you hear that question, Regina?
Yeah, that's definitely something that we've thought about, just like differences in different populations about even for maize, but also teosinte. And that would like probably make sense, especially early on in domestication, for there to be variable selection if multiple populations are being cultivated. And that's just something I didn't take into account with my simulations, where it just like doesn't assume any population structure and it's just one giant population.
Have any effect on germination? The fact that it's not enclosed with this hard cover?
Was the question?
Yeah, how does tga1 affect, or the presence or absence of fruitcase affect germination?
I don't know a ton about that, but I think there's been some connection of the fruitcase or like related tissues to dormancy, and I don't really understand dormancy that much, but potentially has an effect.
And there's a question from Ruben in the chat, whether there are any signs of linked selection between tga1 and tb1.
I am not really sure. I haven't looked into that very much.
My guess, Ruben, is no, since they're on different chromosomes. But they are related in terms of, I believe TB1 regulates TGA1. Somebody can correct me if I'm wrong here. But I don't think there's evidence of selection on sort of both simultaneously, or for some, or for a haplotype, or for for individuals that carry both mutations. I don't think there's any evidence for that. And then Buckler Lab is asking, is the fact you find the maize allele as a heterozygote just a function of allele frequency, or do you think it's selection against the maize allele in teosinte because of dominance, for example?
My personal suspicion is that, thinking about the collection of these seeds, was people collecting what they wanted to be like pure teosinte probably. And if you see an exposed kernel, the assumption is usually that it has past maize gene flow that is causing that maize-like phenotype. And I just think that they probably wouldn't have been collected as much. So if it looked super exposed and would have been, like homozygous for the maize allele, I just think it wouldn't have been collected for this purpose. So that's why I think we're finding like these sneaky heterozygotes.
And there is some evidence, I think, and Michelle has tracked this down well too, from the teosinte hunts in the early 70s, that they did identify teosinte with exposed kernels, and sort of wrote those off as gene flow, and so didn't assume that couldn't be mutations in actual teosinte. So I think that's consistent with Regina's argument.
But the famous Beetle expedition, they didn't find any exposed kernels, right?
No, that's what I'm saying. They did.
Oh, they did?
They were written. They did, but they were written off as recent gene flow from maize, and so maybe those were recent, BC1s or something like that, but they weren't considered to be quote unquote true teosinte.
Okay, I got that story wrong.
Well, that's generally the that's how the story is told, is that they didn't discover it, but there's some bias there because the ones that they did find, they wrote off. That's right. Yeah. So they did do progeny tests on some of these, and so the thought was that it was introgression based on progeny tests. Additional questions? Okay, if not, let's thank Regina again for a cool talk on a neat domestication trait, and we'll be back next week. We're doing a bit of rearranging the schedule, but we will have a talk next week. So we will see you same time, same place. Thanks all.